Good. So genetics was mentioned a couple of times in the past half hour, and James has a question here. Actually, it's more two questions, but I'm just going to read it in full. So he says, I'm taking this class in conjunction with a genetics class, and I was curious how much the advancements in gene sequencing are affecting your field. Do you think many new illnesses will be treatable using genetic procedures such as gene therapy or antiviral immunization made in a lab at the genetic level?
Yes.
Next question. Can you elaborate?
I was going to let other people chime in because I could talk about this for a long time.
That is you. It's definitely your expertise.
It's a fascinating time to be studying any type of disease. So the example I think of are using gene sequence data, and this is whole genome sequencing data, is probably most well documented for cancer therapies, where very simple things like the understanding the genetics of in an individual on how drugs are metabolized means that we can dose patients correctly, okay, and maintain drug levels, and it's making a huge difference in the responses, because either drugs are not cleared as fast or they may be moved into tissues where they're not effective against the cancer. So definitively, yes. I think there's also some wonderful work in cancer therapies where certain types of cancers that are very resistant to treatment, if you have the right genotype, they can be treated now in fairly remarkable recoveries. But in related to infectious disease, I mean, we had this discussion on what constitutes good, effective, long-term immunity. And that's genetically determined. It's not always genetically determined because the environment in which those cells are proliferating are also important. But there's a large genetic component to that as well. And I suspect that it's not an easy problem to solve, but that there'll be quite a bit of emphasis in this area I mean, there is right now, but we'll actually have some definitive information in the very near future on this.
Another example where you can really see genetics being incredibly powerful are infectious diseases of plants, because if it is true that it's genetically determined, which it is, you can genetically modify the plant and give it a very, very robust immunity. And we're seeing this particularly with a technology which is called RNAi, which is RNA interference. So essentially a little blocker which will prevent the parasite, be it a virus or a fungus or a nematode worm, doing its business by the plant expressing this little molecular switch which goes in and stops the parasite reproducing or transmitting. And that's an incredibly effective technology which is wonderful for its ability to be very precise and only target that parasite. So there's a really great example where we could genetically engineer immunity, but in plants rather than animals.
No, I think animals as well. Some of the first use of gene therapy, actually, viruses, of course, remember, they're obligate intercellular parasites, and even for viruses that don't have a good persistence mechanism, they can bring in a gene with them that a host may need. So some of the neurological diseases which are due to the absence of an essential protein or some functional aspect that's missing, we can introduce the gene using viruses or virus vectors. So that's an old technology. I think somebody was discussing as well, why don't we use viruses to fight other diseases? And of course we do use viruses in cancer therapies as well, because one of the responses of a cell that's infected is to kill itself, is to undergo apoptosis, and that's exactly what you want to happen for a cancer cell. So these are not new therapies, but they'll probably become a bit more sophisticated as we learn a little bit more about the genetic mechanisms.
I guess another thing that's another bit of the technology that's revolutionizing the field quite a bit is that there's certainly a whole large number of both viruses and infective bacteria that we kind of didn't even know existed, because they were bacteria that maybe couldn't be cultured before, and so you know, the massive opportunities to gene sequence everything and find everything to do with gene exploration is just shown that, you know, the world of infectious diseases is, you know, 10 times as large as we used to think it was. So.